So good morning, everyone. So my name is Yijie Wang. I'm the associate professor in the computer science department at IU Bloomington in United States. Today, I'm going to talk about our work using physical transcription factor proximity to improve gene regulation inference. So as we all know, gene regulation inference is a long-term challenge in system biology. And in this paper, we'll focus on transcriptional gene regulation. And the ultimate goal is actually to predict the bipartite graph, the gene regulatory network. We have two kinds of genes. And on the top right of this slide, basically, I give a very short, brief illustration of how gene regulation works. So basically, we have the genome, and we have all kinds of genes. Some of the genes are transcription factor genes, some of the genes are target genes. And those genes will be transcribed into messenger RNA and further translated into proteins. And for this transcription factor gene, they will become transcription factor proteins, and those proteins will bind into the genome on the genome to regulate downstream genes expression. And in the bottom right, basically, I listed the things that we can measure along this gene regulation process. As you can see, we can measure the gene expression, we can measure chromatin accessibility, we can measure the motif of those for most of the transcription factors. And one thing you may notice that, so along those measurements just provide the partial information for the whole process of gene regulation. And what we're trying to do is trying to utilize this partial information to reverse engineer gene regulation. So that's make it really challenging. And let's look at the state of the art. So in the past few years, lots of great methods has been proposed. They publish really good journals. They utilize the single-cell RNA-seq data, single-cell attack-seq data, and motif data to do gene regulation inference. And we are doing something similar. So then how it works? So basically, the first thing they do is utilize the single-cell attack-seq data. So what we can find the open region around the target gene. As you can see, we have the gene, that the yellow part is the target gene, and we can check in the open region around it. Like C1 is one open region, CI plus 1 is another open region, and the CI plus K is another open region. And we do we know that's open region, we also know the sequence of those open regions. Okay, and then based on that, then we have, on top of that, we have the transcription factor motif data. So basically, based on those data, we can basically try to identify the transcription factor that have the potential to binding to those open regions based on the motif sequence similarity. As you can see here in the first part of this open region, we have the sequence of TCGAT, and we know the motif for TF1 and TF2. That also TCGAT basically indicates that those two transcription factors could have the potential to binding to those open regions. Then we all go across all those open regions to such motif matching, then we can get a bunch of transcription factor that has a potential binding to those open regions. And but we all know that motif matching does not necessarily actually binding there. We need additional information. So here we have, on top of that, we have the gene expression data. So for example, we know the expression of the target gene, we know the expression for all the transcription factors, it's expression. So if we're using Y to represent the expression of the target gene, using X1 to Xn to represent the expression of all the transcription factors, and all those state-of-the-art method, basically they're trying to do it just trying to find a model, a function between Y and X1 to Xn. Some of the functions are linear models, some of the functions are nonlinear models. And to give a very simple example here, so here, we have 3 transcription factors. We know there's gene expression, we know the target gene expression, they can build a linear model. And based on linear model, we see that X3 has nothing to do with this linear model. So basically, we can infer that X3, the corresponding transcription factor TF3, is not regulating this target gene. Okay, but if you look at all those methods, there's one thing that they're missing to consider is what our group called the transcription factor proximity. So then what is transcription factor proximity? So here I borrow a figure from the biology textbook, undergraduate biology textbook, introducing gene regulation. As you see here, we have the target gene in the green part, and we have the genome band over. We have a transcription factor binding to the promoter region, some transcription factor enhancer region, and then somehow they fold this protein complex and this machine protein complex will initiate the transcription. And also, in recent Nature review paper, they also introduced the same mechanisms. The promoter region and enhancer region, some transcription factor will binding over there, and they were trying to recruit mediators, and they can form a protein complex to initiate the transcription. And based on all these two figures, one thing we can conclude that if several transcription factor they're trying to regulate the downstream gene, then they have to be in close physical proximity because they're folding in this protein complex. They are physically close. Okay, and actually this is not new. And but in the current people doing this, they all know this such a phenomenon. But in all these current methods published in rigorous journals, they don't consider such a thing. So we're trying to consider that. So before we consider propose our model, we do actually trying to using the existing data to test whether this is the true. So basically the things we're trying to prove that is true transcription factor for a target gene are in closer proximity than the random transcription factors. So in order to do that, we need to first compute to figure out how to compute the TI proximity. So basically we compute that on the protein. For example, we have a protein-protein interaction and we have two transcription factors. We can using the shortest path distance to estimate the TF proximity. For example, if you see here, the shortest path is 3, so we can see that the proximity between TF1 and 2 is 3. And also we have other distance that metrics we can use. We have a diffusion state distance. But then based on this, we know how we can compute the transcription factor proximity. Then we using Lutenize the data, we get some PPI network from Belgrade, we gather all the interaction for human and all the physical interactions. Then we have our PPI network, and for the true transcription factors, we get from the well-known database. Then we do the following experiments. And here first we're using shortest path as the distance metric, as the metric to measure the TF proximity. And we have 3 histogram. So first histogram is in red, basically how we get it. So basically we go to the trust database. We're finding two transcription factors. If we know these 2 transcription factors regulate the same target gene, we compute their shortest path in the PPI network. Then we go to find another 2 transcription factor. We know they regulate the same gene, we compute this shortest path distance. Then we go over all those known transcription factor regulate the same gene, we compute the shortest path distance. Then we can draw the distribution of it. And based on this figure, we can conclude that here, the average distance between 2 transcription factors, we know they regulate the same target gene. The distance the average distance is 2. Okay. And this in the middle, basically we change another database, the database providing the true transcription factor, but we have the same observation. Basically, this indicates the average distance between 2 transcription factor, showing this RegNet. Basically, the average distance is actually 2.5 something. And on the purple figure basically shows the background distribution. So basically we go through all transcription factor, pairwise transcription factor, compute their shortest path distance on the PPI network. As you see, this indicates the average distance between 2 transcription factor on the PPI network is actually 4. So this clearly indicates that if 2 transcription factor, they're trying to regulate the same target gene, they have to be physically close. And then we're using another metric, the diffusion state metric, to compute to estimate the TI proximity. We have the same observations. As you see, the blue unknown TFs, the true TFs, yellow are the random TFs. And also you can see that, if the 2 transcription factors regulate the same gene, target gene, their distance is smaller than the regular random selected transcription factors. Okay. And the next thing is how we can design a model, incorporate such information into a computational model. It has not been done before. So and then here we propose our model GRAPE. So basically, yes, it looks like this. Basically have 2 parts. The first part is just sparse linear model. And let's recall Y is represent the expression of the target gene, and X1 to Xn represent the expression of the transcription factor candidates. And we're trying to build a linear model, and we're trying to use an L0 norm to induce sparsity and then to identify the most important K important transcription factors for the target gene. And the definition of L0 norm is like here. And you may ask why we're using L0 norm. So because based on in the past few years, my group did a series of work, basically showing that using L0 norm is better than using L1 norm in the lasso model, in the group lasso model, in the field lasso model, and the generalized field lasso model when the sample size is small. And we published series of good papers over there. But this is nothing fancy, just a simple sparse linear model. So the part that incorporates the t-hat proximity actually is the second part of the model. So basically this part computes the distance between the k-selected transcription factors. And Sij is the diffusion state distance we use. And here I want to emphasize the use of L0 norm in this term. So the reason we want to use L0 norm, we're trying to avoid the inference of the magnitude of the coefficient beta. So no matter beta I is large or small, positive, negative, as long as it's non-zero, we will consider the distance. We will consider the distance to the corresponding transcription factors. So that's very important to use L0 norm. All the existing regularization term cannot achieve that. Okay, and then if overall you look at the whole model, what we're trying to do is very simple. We're trying to find the K transcription factor whose expression can better explain the expression of the target gene, and those transcription factors should be physically close. This is basically the goal of this model. But because we're using L0 norm, the whole thing is hard to solve. But we have a beautiful framework to solve it. So here is how we solve it. First, we just rewrite the problem into the matrix format. And the first thing we do exactly Boolean re-representation. So basically, we have two formulations for the same problem, and they are mathematically equivalent. And after doing some analysis for the Boolean representation, we know this is a convex Boolean program. So basically, the object function is convex. The only variable is a Boolean variable. And basically, it have really good properties. But solving it is equivalent hard to solving the original problem because they are mathematical equivalent. But here we propose to using Boolean relaxation to solve it. So original UI is either 0 or 1, but here we relax it to between 0 or 1. Typically, when we do such relaxation, we're basically changing the problem and most of the time, the solution of the relaxed problem have nothing to do with the original problem. But the beautiful thing of our formulation, actually, we prove that our formulation is tight relaxation. So basically, under certain condition, even we solve the relaxed problem, we can get the solution of the original problem. Okay, then we feel very comfortable just solving the relaxed problem, and this is a convex programming problem, have a unique solution. Then we just using the off-the-shelf algorithm. We're using Project Cozen-Newton to solve it. So right now we have a beautiful model. We have a beautiful algorithm to solve it, and let's just test the performance. Okay, so we're trying to compare our algorithm to the state of the art in terms of in terms of the predictive power of the transcription factor. Based on previous study, we know if the transcription factor are the true transcription factor, you should have the power to predict the expression of the target gene. Okay, then what we're trying to do here, so for the same target gene, different method will output different transcription factors. We're using those transcription factors to build like the similar model, the same like a linear model on the training data set and test their performance on the test data set. We're using the out-of sample MSE as the metric to measure the predictive power. And the data we're using here, we're using PBMC data, so which include both RNA-seq, single-cell RNA-seq and single-cell attack-seq. And we're trying to do gene regular inference for 3 cell types, CD4, CD8, and CD14. And those are the methods I'm trying to compare. And here is the result. And yeah, each column representing each cell type, each row representing the cell type, each column representing a pairwise comparison across the cell type. And each dot in this figure representing a target gene. The X coordinate for that is out-of sample MSE achieved by our method. The Y coordinate is out-of sample MSE achieved by the competing method. So if you see, we cannot see it. Basically, this top left corner is the shade part. So if the point fall in this part, basically indicate that our method achieve better performance than the competing method. And if you look at all those pairwise comparison, you will see the majority of a point is fell into this upper right left corners, which basically indicate our method outperforms the competing method. And the second thing we're trying to compare the prediction, the consistency of the prediction, whether the prediction is consistent with the PC-HiC data. So PC-HiC data basically providing the region, the promoter region interact, we identify some regions that interact with the promoter regions. And here, basically, if we find a transcription factor that is able to binding to the region that have interaction to the promoter region, we think that's a hit. And based on that, we define F1 score to do such comparison. And for the detail, you can look at into the paper. But here is the result. And similar thing here, each dot is representing a target gene. And the X coordinate is the F1 score achieved by our method. Y coordinate is the F1 score achieved by the competing method. But this time we should look at the bottom right part. If the point is falling in this part, basically indicate our method is achieve higher F1 score than the competing method. And if you take a look, the majority of the point is falling in that corner. And basically indicate our method achieve better F1 score in this sense, okay? And yeah, and as a summary, so basically we first prove that using the existing data, prove that if transcription factors, they're trying to regulate the target genes, they have to be in physical closeness. And the second, we propose the very first model explicitly incorporates this TI proximity into the model. And we also believe this model can be used for other bioinformatic applications. And at the end, I want to thank my graduate students involved in this project, and my lab and my collaborator, and the funding resource support the research. And I have one postdoc openings. If you want to do some traditional hardcore machine learning stuff, and trying to apply this to solve challenging biological problems, please talk to me. And thank you so much.
The talk, we have time for one question.
I was wondering if you performed any ablation analysis, let's say removing that extra factor in the objective function, and also how did you set the hyperparameter K?
So basically, yeah. So we actually did the ablation study. It's in the paper. You can find that. And basically, we show that consider this is not for all of them. For the majority of for the target gene, we have better performance in predicting their transcription factors. And how we select the K, basically, yeah, we using, yeah, when the training data set, we just splitting the training data set into two parts, basically in the cross-validation style to select the K.
Thank you so much again for the talk.
Yeah, thank you.